I am so delighted to talk to probably my favorite geneticist in the Chicago area. Dr. William Oles is the science officer for Protein Tech, a Chicago-based antibody manufacturer. But more importantly, you are, in fact, a geneticist, and we want to talk a little bit today about CRISPR.
CRISPR is a genetic technology that was discovered a few years ago in bacteria. It's kind of like the immune system for bacteria.
The CRISPR-Cas9 system is called gene editing. And when inserted into human cells, it works the same way it does in bacteria. Sort of like the select and delete function on a word processor. The Cas9 molecule is fitted with a section of RNA that exactly matches the DNA sequence it's engineered to remove. It's also paired with guide RNA. That, like a microbiological Google Maps, directs Cas9 with great precision to the desired location on the chromosome. Once it arrives, Cas9 uses its enzymatic scissor power to cut the offending DNA sequence from its chromosomal moorings. And Bob's your uncle. Bad DNA be gone. The reason we're talking, though, is that there was big news in the last couple of weeks, reported in the journal Nature, that for the first time, the DNA of a human embryo was manipulated using CRISPR to essentially knock out a genetically based disease. What was done that was such big international news?
It was a gene called MYBPC3. So it causes hypertrophic cardiomyopathy, sudden heart failure. It happens a lot for young athletes, which is quite scary. You could be on the football field and this would happen. It would be really terrifying. They were able to find someone who would donate his sperm. This guy has this mutation. He's a carrier. So he has one normal copy, one bad copy.
You have to have two copies of the bad gene to get the condition. And if you just have one copy of the good, one copy of the bad.
Yeah, so as long as you have one copy, you're A-okay. Then they had donated oocytes from wild-type women.
Oocytes?
Okay.
Oocyte?
The oocyte is what the female produces that becomes the egg.
Okay.
Now we have the female always bringing a wild-type copy of this gene, and the male bringing 50% bad copies and 50% good copies. Now, in this CRISPR experiment, they really only have to fix one of these chromosomes, because in the embryo, they only have one mutant and one good copy.
Oh, so this is really early in the development. This is like really just as the sperm and egg get together. Okay.
Exactly. Now, in this instance, they just wanted to show that they could change the sequence in a human embryo. The key thing here is really the palate, the human embryo.
One of the really interesting / terrifying things about CRISPR technology is, as I understand it, anybody could do it. There are companies that for $150 will edit the DNA in whatever way you would like. If you'd like to have a green fluorescent rabbit, I know that's old news. Any knucklehead with $150 can come up with some novel gene sequence, and the CRISPR folks and will send it off and they'll do it.
There are some companies that are currently available, like Editas.
Odin?
That's another one. So those both have patents, and so they could do it. But there are restrictions in terms of how much DNA you can plop in there.
Once these technologies are out there in the wild, they can regulate it. Marijuana has been illegal for most of my life. It has not stopped its easy availability. So is it really reasonable to expect that people who want to have the ability to do these kind of manipulations won't?
No, I think it's impossible to really prevent people. All you can do is mitigate it.
What should we lay people know about gene editing? How should we think about it?
Personal education, just learning more about it. CRISPR is such a foundational and critical technology that there's lots of videos, podcasts on this technology so you can understand the basics. And the other thing is also to, if you have anyone that you can ask about this, any scientists that you know, like you. Thankfully, you know me, so you can ask me. What can be done and what can't be done? Because sometimes our imaginations are scarier than the reality. This isn't Gattaca or any other science fiction quite yet.
Are you worried?
No, I'm not worried.
Even though it's a technology you think is completely non-controllable, and even if it were controllable, the laws have not caught up with the technology. Why are you not worried?
So the technology is not quite there yet to the point where I would really be concerned about it. Frankly, I think there are better things to worry about at this stage in time.
Well, Dr. Olds, if you say so, I'm not going to be too terrified yet, but I sure am grateful to you for spending some time with. Maybe you can come down to C2ST and give us a little chat with our audience and let them ask you some questions about this. Any chance?
Yeah, that'd be great, yeah.
All right. Well, Dr. William Olds of Protein Tech, thank you so much, my brother. Look forward to talking to you soon.
Likewise. It's always good to see your smiling face there, Aaron.
Right.